The effect of methionine-restricted diet on the mTOR pathway gene expression and telomere length on yeast. Restless leg syndrome, otherwise known as RLS, is a neurological disorder characterized by throbbing and uncontrollable muscle movement or activity in the body. In 2009, RLS has been shown to affect 5 to 10% of Americans. The condition causes pain, sleep deprivation, inflammation, and stress, which all causes the aging process in an organism to increase and quicken. However, having an amino acid restricted diet, more specifically a methionine restricted diet, may lead to a slower aging process of an organism. This diagram shows the methionine metabolism process, where having methionine in the diet can cause faster DNA synthesis to occur, therefore shortening the length of the DNA, which indicates further aging. The caps that are responsible for the shortening of DNA or chromosome length are known as telomeres. These tips initially protect the DNA and they keep everything intact. However, as time goes on and cells reproduce, telomeres become shorter and shorter until reproduction process cannot be continued because the telomeres are no longer present. As cellular reproduction slows down and then stops, we see signs of aging. Yeast have been highly studied as model organisms to better understand aging through genes and cellular pathways. The limited amount of glucose, or possibly limiting the amount of amino acids in the growth medium of yeast, have been shown to decrease RLS and involuntary activity in yeast. Overexpression of certain genes, such as mTOR, has been shown to increase RLS by causing the accumulation of DNA, which is thought to be one of the causes of aging in yeast. These effects may be the result of an increased signaling in the mTOR cellular pathway. Decreased TOR activity has been found to extend aging in yeast. The mTOR pathway involves a protein kinase that regulates cell growth, cell proliferation, protein synthesis, and transcription, so overexpression of mTOR shows RLS and aging. In 2004, Ruckenstuhl performed a study where he reduced the supply of methionine in yeast and tested for the protein expression of ATP synthase in order to check the activity inside the yeast. The results showed an overexpression of ATP synthase protein. Therefore, it showed an anti-aging pathway in the yeast that was triggered by the restriction of the methionine in the diet. Therefore, the results showed that methionine decreased the aging in yeast. In 2013, Lee performed the study Methionine Restriction Extends Lifespan of Drosophila melanogaster under conditions of low amino acid status, and she tested this by observing the longevity of the fruit fly under the restrictions in the diet. Results showed how methionine restrictions in Drosophila mimics the effect of dietary restrictions and is associated with decreased reproduction. However, under conditions of high amino acid status, methionine restriction is ineffective and the trade-off between longevity and the reproduction is not observed. In 2007, Roberts performed a study where she tested the differences in the lifespan of Drosophila by feeding the organism a chemically defined diet with limited amounts of glucose. By observing the differences in the means of their lifespan of the Drosophila, Roberts concluded that the flies with the glucose and the amino acid restricted diet survived for a longer period of time versus the flies with the normal diet. The purpose of this experiment is to test the effects of methionine-restricted diet on a molecular level by observing the expression of the mTOR gene and determining the length of the telomeres. The alternate hypothesis states that the methionine-restricted growth medium will cause the mTOR gene expression to be downregulated and the telomere length will be longer in the yeast compared to the mTOR expression and the telomere length in the yeast on the regular growth medium. The null hypothesis states that the methionine-restricted growth medium will have no effect on the mTOR gene expression and the telomere length. In this experiment, the yeast will be cultured in a liquid culture medium in which the total volume of yeast will separate into two separate groups, one control group and one group with the methionine-restricted growth medium. That group will branch off into different concentrations of the methionine restrictions, the concentrations being 0.01, 0.1, and 1% of the total solution. Then the control group and the variable group will be cultured in the growth medium for 24 hours before performing the procedures. To find the expression of the mTOR gene, there is a performance of RNA isolation, RT-PCR, and gel electrophoresis. To find the length of the telomere, there is a performance of DNA isolation, real-time PCR, and then the DNA strand is tagged with the 36B4 oligomer to find the repeating subunits at the end of the DNA strand. All of this data would be analyzed using a one-way ANOVA post-hoc Scheffé test with a significance level less than 0.05. This slide shows a diagram of the budget of this experiment, with most materials that are easily purchased at Carolina Biological, Sigma Aldrich, or Invitrogen.